means color, toma means layer, graphy means representation. This is the complete representation of separated components based upon its color. So now there would be a fundamental question whether every component has color. No. Then why you call it as a chromatography? Let us see the introduction and its wide application of chromatography. So the term chromatography, was initially coined by Michael Sweet. He separated some pigments onto the column. In doing so, he found that there is a separation technique which separates the pigment components based upon its color. That's why he coined the term chromatography. However, you can separate colorless components also by chromatography. However, you should identify them by other techniques such as visualization by UV chamber, visualization by some other detecting techniques, either by fluorescence or by any other way of identification. Further, if you see, chromatography involves separation of two components, that is equilibration between stationary phase and mobile phase. What do you mean by equilibration? Suppose if a component, you see this slide, in this you have a stationary phase, that means which is stationary, which will not move, and other one is you have a mobile phase which continuously moves onto the column. Now your analyte will be partitioned between stationary phase and mobile phase. If its distribution is more in the mobile phase, it will be eluted. If its distribution is more onto the stationary phase, it will get retained. This is the fundamental definition of chromatography. Now, what components will be retained onto the stationary phase and what components will be eluted from the stationary phase depends upon the polarity of stationary phase and polarity of your mobile phase. And now the first most fundamental in this identification is like dissolves in like. Suppose if you use a polar stationary phase and if you have a non-polar mobile phase, now if you are having a non-polar analyte, now your all non-polar components will be eluted first and all the polar components will be retained onto the stationary phase. Now reversely, if you have a non-polar stationary phase and you have a polar mobile phase, now all your polar components will dissolve in the polar mobile phase and will get eluted. So that means like dissolves in like. So your mobile phase is polar, if your and analyte is polar and you elute polar components, such type of techniques is called as RP-HPLC. What do you mean by RP? Reverse phase HPLC. In the reverse phase, your stationary phase is non-polar. So that is the fundamental definition between normal phase chromatography and reverse phase chromatography. Now, the entire chromatography changes from the beginning to till date is applicable based upon one equation called as Van Demeter equation. It tells you how the decrease in particle size, increase in velocity of the mobile phase helps in separation. So it has a Van Demeter equation, H is equal to A plus B by mu plus C mu, where mu is your velocity of mobile phase and A is your eddy diffusion and B and C are constants. So this will tell you the distribution coefficient of your component between mobile phase and stationary phase and how well they are eluted. Now, plate height. So this is the parameter by which we characterize the efficiency of separation. Now, if you see in any Indian pharmacopoeia or United States pharmacopoeia or any British pharmacopoeia, they give the separation efficiency based upon HTEP, height equivalent to theoretical plates, HETP. So based upon those, characteristics, we characterize the separation process which is better. Now, number of theoretical plates increases, your separation and resolution increases. So this is a fundamental definition of by using Van de Meter equation. Further, now in the columns, there are different types of columns you use. So either it may be capillary columns, preparative columns, analytical columns. So it depends upon what type of technique you are going to use. Suppose if you want to use a preparative column where your sample load is very high, that means the column diameter as well as this internal diameter and external length will be very high. Suppose your analytical column where you want to separate your components, again your column length will be reduced, and your capillary and nano columns will have very smaller in size. Now further, this is with respect to diameter. Now with respect to particle size of your stationary phase, that is your silica or any, you can use different types of stationary phase, pellicular silica. Nowadays you have different types of stationary phase available in the market. However, the particle size of the stationary phase will determine whether your elution technique is HPLC or UPLC. What does it mean? HPLC terms that it is high performance liquid chromatography. Now if you go for a UPLC, that is ultra pressure ultra performance liquid chromatography. What does it the difference between HPLC and UPLC? The HPLC, the pressure applied is up to 400 bars, whereas in UPLC the maximum pressure applied onto the system is 4000 bars. That means a 10-fold difference in applied pressure. What it will tell you, as the more pressure, your stationary phase should have a capability of withholding the pressure applied onto the column. So your particle size should be smaller in nature. So the lower the particle size, high pressure of withstanding the high pressure onto the column, so faster is the elution. So this is the fundamental definition which you need to know in elution technique. Now coming to an HPLC instrument, how it looks. Now if you see a schematic representation of an HPLC where you have two solvent reservoirs, so that means the pump is your binary. Suppose if you have four solvent reservoirs, you have even quaternary pumps. Now here it is a controller which controls the flow of mobile phase. Now based upon this controller, I can fix that if pump A can pump 100% of the solution, pump B 0. What does it mean? So completely only from one mobile phase I am using in the system. So that is called as isocratic elution. Now whereas by using the controller, I will set the system in such a way that 40% of mobile phase A and 60% of B will run throughout the column. What does it mean? Even here, I am fixing that 40 and 60 ratio by using binary pump. I am pumping through the system. Even now, it is an isocratic system. Now, what is gradient? Gradient means initially I pump only 100% of mobile phase A, and step by step I will add my mobile phase B. In doing so, I will see what type of changes the incoming analyte is happening onto the columns as well as onto the elution process. This step is called as gradient elution. What does it mean is that I am continuously passing one mobile phase and step by step I am adding the other mobile phase into that one. This type of technique is called as gradient elution. But in gradient, again you have two types. One is linear gradient and another one is step gradient. What do you mean by linear? Linearly, but by programming way, linearly I am increasing the mobile phase B. Whereas step gradient, I do not follow linearity. I just increase 10%, 40%, 70% abruptly. So such technique is called as step gradient. So this is a fundamental differentiation between isocratic system and gradient system, which you need to know. Now what we have, vacuum degas. This is the most important in the HPLC system. So before you use any mobile phase, that should be filtered by either by 0.45 micron filters or 0.22 micron filters. And after filtering it, you should follow vacuum degassing or by simple sonication method. What we are doing, by doing all these things. In doing all these things, we are removing the dissolved gases from the system. So when the dissolved gases are removed, the mobile phase, it will flow with an uniformity throughout the system. So that is the fundamental information which you are getting from using vacuum degasser. Now what is binary pump? Binary pumps, again you have pumps, there are different types of pumps. One pump is you have a reciprocating pump, pulsating pumps. So again the definition and the working principles of pumps you can go and read on to any book. So I am not going to deeply about the pumps. So these pumps will pump the mobile phase based upon the programming you have provided here. Next comes the auto sampler. What is auto sampler? This will pump injection either by two types. One is by manual injection or by auto sampler. Auto sampler uses robotic techniques. Now whereas manual injection uses rheodine injectors. So if you can see here, there is a symbol of injecting loop where you inject using your Hamilton syringe. So now your sample is in either in load position or in inject position. Based upon the shifting the valve, you will allow the component to flow into the stationary phase column and followed by the detector. Now your auto sample samples the programmed auto sampler. Further it will go on to the column and column. See here it has shown that a thermostated column department. What does it mean? Suppose if your elution without heating the column, you are not getting proper elution. You may get tailing or you may get fronting onto your chromatographic peaks. So to avoid that one, we have a provision of column oven. However, the temperature of column oven cannot be increased more than 80 degrees centigrade. So maximum up to 80 degrees centigrade you can use your column oven. So based upon that you should use different types of silica which can withstand higher temperature. So that is also a very costliest method. Followed by, once you program with the temperature, whatever the tailing and fronting which you are getting with the signal, you will be processed in this method. Further, it will go on to detector. What is detector? It will identify the analyte concentration based upon the input mobile phase as well as analyte. So now you will get a signal onto your processing unit, that is your computer, and you will get the information on based upon total number of components. So this is the fundamental definition of an HPLC. So we will see by an animation how an HPLC works. So this one is your animation. Let me show you. So what's happening here? Okay. Now when see, I'm injecting a sample. Now your elution is based upon what components are retained and what components are eluted. So your chromatogram is displayed based upon. See, now if you have a chromatogram signal far behind the retention time is very high. What does it mean? This component is retained more onto the column and this component is retained very less onto the column. This is the fundamental definition. And here you are using two solvents and two pumps. There is a binary pumps, and here is a mixing unit, and it will go on to the detector. This is the fundamental definition of your separation. Now let's see what. Now we will come to GC. Based upon the same principle, we have found a newer technique that is called as gas chromatography. Now gas chromatography here, irrespective of your mobile phase, which was liquid in your HPLC, here it is gas. That is, your mobile phase is gas and your stationary phase is either liquid or solid. This is the fundamental information of gas chromatography. Now, as the term chromatography was coined by Michael Swick, and the term gas chromatography was invented by Martin and Sinch in 1952. This is the information you need to know. Further, the types of stationary phase in gas chromatography are several types. Suppose that is a wall coated open tubular column, or surface coated open tubular column, or particular coated open tubular columns. This tells you whether your stationary phase is solid or liquid. What do you mean by solid stationary phase? That means you have a particles fitted onto the stationary phase that may be either capillary or that may be packed columns. Now the next one is your stationary phase is surrounded by some amount of liquid system coated onto the stationary phase. The coating onto the stationary phase is called as, your stationary phase is now here liquid. So based upon that, whether your stationary phase is solid or liquid, you will determine the principle of separation. Suppose if your stationary phase is solid, now the principle of separation is adsorption. If your stationary phase is liquid, the principle of separation is partition. So this is the fundamental information you need to know whether your stationary phase is solid, whether your stationary phase is liquid. However, the mobile phase is continuously, it is gas. What type of gas? You use nitrogen, helium, argon. So there are different types of gases used in system. So if you see, these are the instrumentation setup. You can see different types of cylinders, gas cylinders here. So you have a pressure regulators which regulates the input flow of the gas, and this is your system of ovens. So that means how it operates. So again, if you see here, you have something called purifiers, gas purifying system. So what they have is generally they have silica or some other material which removes unnecessary moisture from the input gases. Now your gases is free from moisture and unwanted things. Now the gas will flow into the system and you will separate. So this is the sampling method in GC. We will see by one of the animations which I will tell you now. So the first animation I will tell you about liquid chromatographic system. So here in the liquid chromatography, when the only mobile phase is flowing into the system, you do not see any type of signal. When the mobile phase is carried with some amount of analyte, you can see the signal. This is the fundamental definition in your chromatography. Let's see with some animation. So see here in the current animation, so only mobile phase is flowing through the system where your chromatogram has no signal, that will have only baseline. Once your mobile phase is carrying some analytes, you see as the analyte is reaching towards the detector, it will show some chromatogram. So here again, if you see, only some portion of the analyte is passed on to the detector system, and much more is released into the waste. What does it mean? That means if you load completely your analyte onto the detector flow cell may be blocked by the incoming analyte. So you should program in such a way that a portion of analyte goes onto the detector and maximum portion of your analyte goes onto the waste. So this is the definition. Now we will see what type of detectors are used. Here the first detector is your UV visible detector that will identify based upon your lambda max of your component, so as well as the absorption principle, that is Beer-Lambert's law. So here as the flow moves through the system, as the chromatogram progress based upon the lambda max. So again there are two types of detectors in UV a simple UV, or sometimes you have a dual wavelength detectors, or sometimes you have a PDE. Now the next detector is your refractive index detector. Now these detectors are termed as universal detectors. What does it mean? The change in refractive index of a system when the mobile phase continuously flows into the system. When the analyte comes to the detector, there is a change in refractive index. The change in refractive index is the principle of analysis in your refractive index detection. So here, see, when the mobile phase is completely passing on to the system, when there is an analyte starts, there is a change in refractive index. That refractive index is characterized by the proportion of analyze in your analysis. However, this is very problematic when you use in gradient HPLC systems, whereas isochromatic HPLC, your refractive index detector is the best detector for your analysis, because any change in solvent drifts, a large change in refractive index causes very bad chromatographic signals in gradient HPLC systems. Further, let us see the other types of detectors which are used in HPLC. Now this one is you can even use the fluorescence detectors as well as phosphorescence detectors based upon the specific characteristics of your components, whether your components are self-fluoresceable or whether it can excite and emit towards an intermediate phosphorescence. So this is based upon Jablonski diagram. So this is the fundamental definition of a fluorescence detector. If your molecule is what do you say, self-excitable or self-fluoresceable molecule, such molecules you can use by fluorescence detectors. Further, if you see the working principles of other kind of detectors, so this is the basic of by analysis using fluorescence technique. The forthcoming way, this one is improving nowadays. This technique is called as evaporative light scattering detectors. This is the fundamental definition which most of the textbooks do not have. What does it mean? The mobile phase which is coming along with the signal, along with the analyte. Now you do not require the mobile phase component onto the detector. If you have a mobile phase component onto the detector, while using your refractive index as a detector, that will cause a broad chromatographic signal based upon changing refractive index of the mobile phase. In order to avoid that, what we are doing is we are going to evaporate the mobile phase before coming to the detector, so that only the component of interest, that is the analyte, will pass on to the detector. So let us see how it happens. So here there is a flow in the mobile phase. Now it is mixed with some nebulizer gas. Now it is heated in a heated zone. In the heated zone, what happens that the mobile phase gets evaporated. Only the analyte, that is red color, the analyte which of interest will go on to the detector. The next coming detector is, this is called electrochemical or redox potential detector, where you have electrodes between mobile phase as well as on the component. When there is change in oxidation or reduction between the analytes, so you have a signal in your chromatograph. So based upon this, you have a detector technique. So this kind of technique is used in where the analytes are based upon the redox potential. So this finishes the basic principles of liquid chromatography. Now let us see the fundamentals of gas chromatography. So see, now this comes the picture of gas chromatography. In the gas chromatography, you have a column oven. So here you have this circular portion which I am showing here is your column. Now columns are again two types. One is a capillary column or another one is a packed column. So this tells you what type of columns you have. Again, you may have stationary phase, whether it is solid or liquid. So you have to see whether it is a silyl or polymethoxysilane and what is the portion of silyl groups. So you can say it is whether it is non-polar or polar, and you have an inlet system where you inject your sample. So based upon my last slide in my based upon my last slide, so here if you see, you use Hamilton syringe to input your sample onto the column where you have a carrier gas as well as that is your mobile phase. So let us see what happens in the system. So here, this is a carrier gas, that is hydrogen or argon or sometimes your helium or nitrogen. This is your mobile phase flowing into the system, and from here you will input your analyte of your interest, and that will go on to the column. Now this is column oven. What you are doing in the column oven? You will program the oven temperature as well as the inlet temperature. These two things you will program in your gas chromatography. Now your input mobile phase, that is your gases, these are generally inert in nature. These will elute the components based upon its ability to either retain or elute onto the stationary phase. You can see onto the detector system. So let us see how it works. Again, the injection mode in gas chromatography is of two types. One is split injection, another one is splitless injection. What do you mean by split injection? When your sample is very rich in analysis, you should follow the split method. In the split method, you will give an criteria that maximum portion of your mobile phase goes through the vent and only the analyte concentration will go on to the detector. So let us see. So now see, the mobile phase is flowing. Now it is going to the outside. When you have an inlet one, now you see this is the inlet with the analyte. You have a lot of analyte here. Now this analysis, if it completely goes on to the detector, there is called detector load or detector cell load, which will completely choke the detector. So such type of techniques are not feasible or not allowed by any chromatographic system. So that's why we use an vent system here, which will differentiate or separate into two different paths where maximum portion of the analyte goes as a waste and only a fraction of analytes goes onto the detector. Let us see. You see, yeah, 90% of the analyte